Good evening, everyone. Let's wait for the students to join, then we'll start with the class. Please just confirm whether the audio and the video is clear or not. Just confirm it in the chat sections. Today, we'll be discussing about the IP3-DAG pathway. Okay. Just check whether the audio and the video is clear to you or not. Okay, thank you, Radhika. Let's wait for everyone to join. So if you haven't yet watched the previous two marathon classes, we discussed about what is cell signaling all about first in the first class, and in the second class yesterday we discussed about the GPCR. Okay, so make sure that you are following all the marathon classes. It would help you out to go through all these things in a better manner. First of all, if you are new to our channel, do not forget to download our application via the App Store and also via the Android. Like it is available both on the Android and also on the iOS, both for Android users and also for the Apple users. You can download the application and get yourself in that particular application, where we have a lot of courses, lot of contents available for you. It is there. The most important one is this particular course, that is the target June 2023 examination, in which we are having on daily basis we are having classes, live classes, where every single thing would be discussed with you, talking about be it the theory class, be it the general aptitude course or the hard copy notes. Everything would be provided to you in the single course at minimal price, where the top concept of installment is also there. That means you can pay via installments and get yourself registered. In addition, we also have the doorstep delivery of the notes booklet now available in all over India. Wherever you are staying, the notes would be delivered to you. As and when you wish to get the notes, you can WhatsApp us on the number and you can have the query. You can have your query resolved for this particular thing and place your orders. Okay. We also have the mock test given to you, which you can prepare very nicely and practice. Talking about today's concept, we have discussed about all of these things in the previous classes. Today we will be discussing about the IP3-DAG pathway. Before that, yesterday we discussed about what is GPCR. Let me give you a quick revision for that matter. So we discussed about the GPCR. What is GPCR? It is G protein coupled receptor. Okay. So over here what happens, you have 7 strands in this particular complete protein, which would be spanning the complete or the entire plasma membrane and helping in the final transduction of the pathway. Okay, now let's understand over here. See, it is this particular pathway, that is the IP3-DAG pathway. It is also called as inositol triphosphate. Like see, I stands for inositol, P stands for phosphate. So because there are 3, so it is IP3. So we call it as inositol triphosphate DAG. DAG stands for diacyl glycerol GPCR pathway. What does this mean? Over here you will be having the usage, or over here we will be getting the concept of GPCR involved. Which are the various ligands which could participate, or which participate via the help of the IP3-DAG pathway? First is the hormone. We can use neurotransmitters, we can use the growth factors, hyperstomia stimuli, for example angiostensin, etc. Now when we'll be discussing about this particular thing, so over here you will be having a lot of abbreviations getting used up. So let's discuss that what is the significance of this pathway first, and then we'll understand this complete process. So talking about the significance of the IP3-DAG pathway. Now see, I am taking very small chunks of information in every class. Why? Because first, to make the things very easy for you, because we have discussed what is GPCR in the previous class, and today we are going ahead with the first component of GPCR, which is the IP3-DAG pathway. So you also do it in the same manner, that you are following the complete sequence so that you understand the things. Because जब हम एक sequential तरीके से concept समझते हैं, so at that particular point हमारे concepts first easily clear होते हैं, and second जो previous year questions हैं या फिर कोई भी आप questions practice करेंगे, you will see that you are able to mark the answers more accurately. So make sure that आप सारे पॉइंट्स को वन बाय वन इजीली मैच डाउन करें. देन टॉकिंग अबाउट द सिग्निफिकेंस. So in this significance, the very first one is the IP3 helps in the mobilization of the calcium 2+ from the storage organelles, and to regulate the cell proliferation and other cellular reactions requiring the calcium 2+. Okay. Next, in the smooth muscles, increased calcium ion concentration helps in the contraction of the muscles. It is also used in the slow block of the polyspermy. In the sea urchin, it is one of the topic of the developmental biology. And last is in the nervous system, the IP3 acts as a secondary messenger, in which the cerebellum have the highest concentration of IP3. Let's understand about this IP3-DAG pathway in the cyclic manner. First thing first, these are my notes itself. These are all handwritten, so it is easier for you to understand. See, we have the GPCR, right? So in GPCR, the ligands come and bind, and over here, if you remember yesterday we discussed that the G protein is having alpha, beta and gamma subunit initially in inactive format, but as and when the GTP comes and binds via the help of guanine exchange factor, what will happen? G protein's alpha subunit gets activated. But depending upon the various like which one would be taking part, so over here what would happen, alpha and beta would be getting alpha would be removed, beta and gamma would be on the other side. So now what would happen, see, activated G protein would be there, so this alpha subunit would be now interacting with what? It would be interacting with PIP beta, PLC beta. So what is it? It is phospholipase C. Just a second. Yeah. Please stop making any kind of nuisance in the chat sections. Do not disturb the students, please. Okay, so we have PLC beta. So sorry. So what is PLC? PLC stand for phospholipase C. So now what happens? This phospholipase beta, PLC beta would be converting, would be activating the PIP2. So this PIP2 would now segregate into DAG and the IP3. That means DAG is diacylglycerol and IP3 is phosphoenol triphosphate. This IP3 would go and bind where? It would go and bind to the ER, that is the smooth endoplasmic reticulum. Remember this particular point. IP3 would be getting bind, or it would be going and binding to the smooth endoplasmic reticulum, जिसकी वजह से जो हमारे calcium 2 plus ion channels हैं, that is the Ca2 plus ion channels which is present inside the smooth endoplasmic reticulum, it would be getting opened, and as a result जो calcium ion channels remove out होंगे, जो calcium वहाँ से remove होगा, it would go and activate the PKC. Now if you remember yesterday we have discussed about what is PKB, what is PKC and everything. Okay, so these are the same things that we were discussing yesterday. Please ignore the messages that are coming on to the chat section. Some people are making nuisance for no reason. Okay, so this this KPC comes and gets activated, and as a result of this, what will happen? The further process, depending upon whatever role it need to play, whatever the functionality need to be done, it gets completed. So PKC, it translocates from the cytosol to the plasma membrane because of the production of DAG. Now it is a kinase, and it would target the serine threonine residues, and as a result of the serine threonine residues, what will happen? The other signaling, the other main signaling pathways which need to get activated, those gets activated, and finally the function which the particular cell need to perform, it performs the same function. So you can see PLC beta, it would be catalyzing the reaction for what? For breaking down the PIP2 into what? Into DAG and IP3. So this is PLC beta. It would catalyze the reaction for converting PIP2, for breaking down the PIP2 into the DAG and the IP3. Okay, DAG would remain sorry, DAG would remain in the cytoplasm, in the plasma membrane itself. It would remain attached to the plasma membrane, and the IP3, it would diffuse out of, what is happening in the chat section? Okay. So now this is the main pathway. What happens initially? The GPCR, via the GPCR the ligand comes and binds. The G proteins alpha subunit gets activated. As a result of that, it would be interacting with PLC beta. So as and when the PLC beta would get activated, it would help in the catalysis of the reaction, where the PIP2 would get converted into DAG, or it would get broken down into DAG and IP3. DAG would remain in the cytoplasm, and IP3 would be diffusing into the cytosol, and finally it would entering the ER, that is the endoplasmic reticulum. And which ER? It would be entering into the smooth endoplasmic reticulum. So now what will happen? The Ca2 plus, the calcium 2 plus channels would come up, they would be getting opened, and as a result of the same, is there any audio issue? Hello. Just confirm whether there is any audio issue. I guess the voice is breaking. It's working fine. Hello, hello, hello. Okay, okay, okay. I guess there was some issue, like I was not able to mark properly. Some students, I don't know why they are doing this way. They're just disturbing the whole class. Okay, never mind. Let's continue. Okay, so we discussed about the GPCR and then the complete process. So now look into this particular thing. You have the PIP2, which stands for the inositodyl phosphate. It is the phospholipid that is present in the plasma membrane. Then we have PLC beta, that is the phospholipase C beta. Over here we have DAG as well, which is getting used up. It is hydrophobic and it is a physiological activator of the pKa-C, okay, and it has higher affinity for the calcium 2+ channels. What will happen? You will be having PLC beta. This PLC beta would be breaking down the PIP2. It would break down the PIP2 into what? Into the DAG and IP3. So this DAG would be remaining in the plasma membrane, and IP3, it would diffuse to the cytosol and enter into the endoplasmic reticulum. Endoplasmic reticulum, the majorly the smooth endoplasmic reticulum where the calcium would come out, and finally the work gets completed. Now we have the PKC, that is the kinase acting on the serine threonine residue that translocates from the cytosol to the plasma membrane, which is used in the production of the DAG. So this is the simple basic main pathway which you need to remember. Just go through this particular pathway. First, very important, and second, time and again questions get framed majorly from this particular area, that which endoplasmic reticulum gets which ion channel gets opened, or second question could be which particular component separates from the plasma membrane and moves to the cytosol for the opening of calcium ion channels. Third, it could be which particular subunit of the G protein would be helping in the breakage or would be helping in the dissociation of the PIP2 into the various subcomponents. Okay, just go through it. If you have any doubt, any problem, please ask. It is written over here, see. DAG is hydrophobic in nature, so it would remain attached to the plasma membrane. And IP3, because yes, it need to transport, it need to move towards us into the cytoplasm, it is hydrophilic in nature, so that it could move or it could reach to that particular position and start the work. Okay, is it clear to you now, Janvi? Is the doubt clear now? Good evening, Kaveri. The concept is almost done. Watch the recording now. It was a very small concept that we had to discuss today, which was regarding the IP3-DAG pathway. Okay, let me give you like you can watch the recordings and get the one and get it completed. Okay, so this was about for the today's class. Just very small topic I have taken up for today. Tomorrow we'll be having the next topic discussed. Okay, so if you wish to have the notes booklet in the hard copy format, you can WhatsApp on this particular number. You'll get all the details, you'll get all the sample content as and when you wish to, and if you have any other query, you can solve it out via the messages. Okay, so this was about it. Let's meet tomorrow, and make sure that you are joining on time. Kaveri, make sure next time you are on time, and other students as well. And please do not all the students, like if anyone, this person who was spamming with some bad messages, make sure that you not repeat this. Please do not disturb the class. Okay, bye everyone, take care.